three major components. Some way of measuring the particle position, a control system, and an output actuator which repositions the particle. Now, the aspect of single particle tracking where there has recently been the most variation between different methods is in the position measurement. So how do we measure the particle position? There are many strategies which can roughly be divided into the categories of camera-based, multi-detector, or patterned illumination approaches. And we focus in this study on the patterned illumination approach. So we considered three commonly used methods, namely the orbital method, the Knight's-Tour method, and min-flux. And so the idea is that in each case, there is some temporal variation in the intensity according to the patterns as shown. And then we extract the position then from the variation in intensity over time. And that we can then get the position. And these methods were designed for different purposes. So yeah, the idea is really just to compare these methods. So all of these methods have mostly been applied using fluorescence, but there's also a method called Icecap. And it is short for interferometric scattering. And it's essentially a different source of photons. And so the idea is that when you have light that is incident on an optical interface, such as a glass microscope slide, the illumination beam will produce both a reflected field from the surface as well as a scattered field from the particle. But then there will also be an interference term, which is indicated as shown. So there will be three terms in your detected intensity. Now, this pure scattering term is going to be negligibly small, but the interference term can actually be large enough to be distinguished from the background. Icecap has some important advantages over fluorescence. Firstly, it can be performed on non-fluorescent samples without having to attach a fluorescent label. Secondly, by using a wavelength where the particle is not strongly absorbed, we can use a much higher illumination intensity without photo damage, and this leads to a more precise measurement. And then thirdly, small fluorescent molecules typically exhibit blinking and photo-bleaching behavior, and this makes it hard to track them once they blink or bleach. And with Icecap, we don't have this problem. Okay, so the big question this study wanted to address is which method is the best? And this comes down to precision and speed. So comparisons are usually made using experimental results, but this is not a good way to compare different methods. And the reason is that samples and setups vary wildly. There are different lasers, different types of detectors, and all sorts of different samples. So we sought to develop a good framework for comparing different methods theoretically and essentially more objectively. And the way we compared it is also extendable to other methods that we have not yet considered. So for this purpose, we had two strategies. Firstly, to examine the statistics of static particle localization, and secondly, to use a dynamic simulation to compare the performance. Firstly, we look at the so-called static localization precision. Now, the idea here is that we ask the following question. Given a finite number of observed photons, how precisely can we measure the particle position when it is not moving? This is important since if the feedback system is fast enough, we can approximate a particle as standing still during one cycle. For this purpose, we use a very powerful statistical tool called the Kramer-Rao bound, which is a lower bound on the variance of any estimator. To elaborate a bit on that, the idea is that we measure the photon counts, and from this, we estimate the position as a function of the photon count. And there can be different ways to do this. So this function is called an estimator, but there can be more than one way of doing it. And we can quantify how good a specific estimator is by its variance. But any estimator that we can find has to have a variance greater than or equal to the Kramer-Rao bound. So that is why this is such a powerful tool. It really tells us, no matter the estimator, uh, what is the best possible performance. Okay, so let's see the results. Starting with fluorescence. So here we assumed a measurement of 100 photons and have plotted the Kramer-Rao bound as a function of the particle position for the orbital method. So two things we see is that the precision is better in the center, so a lower variance in the center. And the other thing is that as we make the tracking area smaller, you see this decrease in the precision, at least in the center. For the Knight-Stuart method, we see a very even precision, so it's almost constant over the scanning range. But again, we see that as we make the scanning range smaller, we get an improved precision. Now for min-flux, we see there is an excellent precision in the middle, around 2 nanometers for a small scanning area. However, as we move outward, the precision gets much worse. We also see that there's a very large variation. As you can see, there's order of magnitude variation as you change the size of the scanning area. So here we have a comparison of all three different methods. What we see is that min-flux has the best precision in the center by a lot, but at the edges, the precision of min-flux is really bad. And also, min-flux has only a good precision when the tracking area is made very small. When you have, as in green, the tracking area is the same size as the orbital method, min-flux actually performs worse. And then we see at the edges of the scanning area, the Knight-Stuart method has the best precision. Okay, so Icecap is a little bit different. And we need to modify our approach here. And the reason is that the uncertainty is, in this case, determined by the background. To see why, we can essentially simplify this because we can neglect the scattering term. We can say that the detected photon counts is approximately equal to the sum of the background and the interference counts. And we actually make this measurement in terms of the contrast between the interference and the background. But because the background is so much larger than the interference, this background is what the uncertainty in the background is what determines uncertainty in the contrast, which is what we actually measure. And therefore, this is also what determines the uncertainty in the precision, in the position measurement. So we had to derive a CRB in a different way than for fluorescence. And what we see is that for the orbital method, whereas with fluorescence, we had a minimum in the center, here we have a maximum in the center. Again, the CRB does decrease with a decreased scanning range, but as a function of position, the CRB actually has a maximum. And the reason for this is a little bit complicated, but essentially it comes down to the fact that because of the uncertainty arising from the background, which is constant, you actually there's actually more uncertainty when a particle is in the center, whereas at the edges, yeah, the precision is actually better. So to compare fluorescence and Icecap with each other, we decided to use a few real-world examples. The examples we used are HIV virus labeled with a quantum dot, a green fluorescent protein, which is an example of a small, highly fluorescent protein, and then LHC2 and phycobilisome, which are a medium and a large light harvesting complex. Now, for each particle, we had to determine the contrast as well as the ratio of Icecap to fluorescence counts. And for this, we used the scattering and absorption cross sections, as well as the fluorescence quantum yield. Um, and so then we can get a result like this, where we have plotted the Kramer-Rao bound as a function of the number of photons. Now, there is only one graph for fluorescence, and the values of the Icecap curves were calculated using the ratio of Icecap to fluorescence counts for each sample. So the displayed counts on the graph are over here are for fluorescence, and then the other curves are the Icecap counts are higher by this the ratio shown here. So we see that for labeled HIV, the precision of Icecap is actually much better than fluorescence. And then for phycobilisome and LHC2 in a micelle, the performance of Icecap and fluorescence is comparable, whereas for LHC2 by itself and GFP, Icecap performs worse than fluorescence. And so this sharp increase we see in the Icecap uncertainty is due to the dominance of the background term in the uncertainty. I will also talk about some dynamical simulations. So here we have simulated the diffusion and plotted the tracking error as a function of the diffusion coefficient. And this is for fluorescence for the 3 different scanning patterns. And what we see is that at slow timescales, min-flux has a better precision, whereas at fast timescales, the Knight's-Tour method is actually able to track the fastest diffusion. And the orbital method is something in between, not as fast, but the same precision as the Knight's-Tour at slower timescales. And this confirms our result from the static analysis, where min-flux has this increased precision when the particle is able to stay in the center because it's diffusing slowly. And because the Knight's-Tour has the best precision at the edges, it is also able to track the fastest particles. Looking at the simulations for Icecap, we see this. Again, there is only one curve for fluorescence. And yeah, we just assume different relative intensities for Icecap. So what we see is that HIV is tracked much better using Icecap, and LHC2 in a micelle and phycobilisome are almost tracked. And in fact, if we double the illumination intensity, both phycobilisome and LHC2 in a micelle become trackable, as shown here. It is important to note that Icecap can be performed at a wavelength where the particle does not strongly absorb. And this means that you can actually use a much higher illumination. So double the intensity is just the start. You can actually increase it much more. So yeah, this is just an example that even just doubling the intensity already makes this precision better. Okay, so some conclusions. Firstly, when comparing the 3 different methods using fluorescence, we find that min-flux is the most precise. However, there is a trade-off with speed. So the smaller tracking area actually means that you cannot track particles that are very fast. The Knight's-Tour method is the fastest because of the larger tracking area. But it is very important to note that both the Knight's-Tour method and min-flux are more costly to implement than the orbital method due to a much faster beam scanning speed that is required. So the winner in terms of precision would be min-flux. In terms of speed, it would be the Knight's-Tour. And in terms of cost, it would be the orbital method. Now for Icecap, we see that the relative success strongly depends on the specific sample you're looking at. And specifically, this is the pigment density and the particle size. So the larger particles tend to be better, and less pigment with less pigments tend to be perform better with Icecap. But increasing the intensity can actually make Icecap a better option. Okay, so what are the key takeaways? Firstly, it's a good idea to compare these essentially experimental methods, to compare them theoretically, as this is a much more objective way of doing this. Secondly, this analysis can easily be extended to other methods that we have not considered yet, since there are quite a few other methods, and you can also extend it, for example, to 3 dimensions. Lastly, IceCat is a very promising um, method, even for samples that are intrinsically fluorescent. Okay, I just want to thank my supervisor, Prof. Todd Kruger, also the NRF, the NLC, the South African Academy for Science and Art, um, and yes, also just to the conference organizers for allowing me to talk. Thank you.
Right, Bertus, thank you very much for an interesting talk. We have about 3 minutes for questions, so the floor is open for questions. Okay, maybe to break the ice, a question from me. So, uh, what is the difference between this type of tracking, the real-time feedback tracking, and the type of single particle tracking that is most often used and based on post-processing?
Yes, a good question. Yeah, so there's a very the most commonly used type of or the thing that is most commonly referred to as single particle tracking is actually just essentially looking at images that are taken over time, and this can be, for example, super resolution images, and then trying to determine the trajectory of a particle um, after the fact. So you just acquire a whole bunch of images, wide field images, and then after the fact track it. But this kind of tracking, we actually follow the particle in real time, and this allows us to at the same time as tracking the particle, also measure the spectrum and the lifetime and all kinds of other spectroscopic properties of the particle. So it's I believe, a more powerful technique in some ways, and also very useful in some ways that post-processing based tracking is not.
Thank you, Bertus. There's a question from Emmanuel. Emmanuel, please go ahead.
Yeah, thanks for a very brilliant presentation. I'm just curious. You mentioned about light harvesting complex 2 in the presence of micelle and without micelle, right?
Yes.
Um, so the micelle means that this is a membrane protein, I would suggest, right? I suppose.
Yes.
So it means you isolated from the membrane while you are using a detergent.
Yes.
And how did you keep the light harvesting complex without the micelle?
Okay, so this is, I mean, this is just a this really these results are purely theoretical. So so I mean, that is actually why I considered the micelle as well, because in any kind of, or in, yeah, almost any kind of experiment, you would have this at least a micelle, or could be a nanodisk or something like that. Um, but so you wouldn't have just LSE2 itself in water. That would not be an experiment. But yeah, I just wanted to, Yeah, so I mean, in terms of applicability to experiment, I think the MyCell result is more applicable. Um, yeah,
All right. Thanks for the clarification.
Okay, thank you very much. We have to cut
yeah,
this short, but if you have more questions, please do write your questions in the chat and the presenter can continue to respond to you during the next talk. Okay, so yeah, let's give the speaker another round of virtual applause. Thank you very much. Okay, the next speaker, I just want to see if she's around. So that is Agnes Mbonyerie-Vuze. I don't see her in the participant list. Lawrence, do you maybe know about something about her?
I don't know. I don't know her, but I don't see her either. I
Yeah,
sent her an email, but I haven't gotten a response.
right. In that case, let's move on to the next speaker. And if this one shows up, we can then just slot her in when she comes.
Yep.
Right. So the next speaker will then be Dr. Abdo Alfiki, if I pronounce that correctly. And
Yes.
that talk will be about fighting against COVID-19, a computational biophysics approach. So I'm sure that topic will be of interest to everyone here. So Epto, you
Yeah.
can share your screen and the
Yeah.
floor.
Oh, thank you very much uh, f- for this invitation, and I will share my screen now.
Yep.
Okay. So first, I have to introduce myself. I am Abdul Fiki, associate professor at Cairo University, working in computational biophysics, and I was junior associate at the ICTP. I have my PhD from Cairo University, 2013, and I was a postdoc fellow in London University and the University of Alberta. So our talk today will be about coronavirus. All of us know about this pandemic disease. So how physics or biophysics can help in fighting this very dangerous virus that spread all around us and make a lot of deaths and a lot of hospitalization and affect our health, our economy, our everyday activity. So COVID-19 is a pandemic. 2.7 million people are dead during this year, and still we have every day new deaths and new infections. COVID-19 was the seventh defined human coronavirus to date. We have six human coronaviruses that we can see here. There's four immersed human coronaviruses, HK1, NL63, and 229E, OC43, that are spread during the last 20 years, but the infection was not as, the strong like SARS or MERS-CoV and SARS-CoV-2. So the most important three types of human coronaviruses are SARS-CoV that mainly affected China and the East Asian countries, and it was about 20 years ago, in 2002. Then come MERS-CoV or Middle East Respiratory Syndrome coronavirus, which affect the Middle East region mainly. And now we have the SARS-CoV-2 pandemic that affect all the world. So the virus spread is different from different region in the world. So in Africa, we have about 4% of the total infections around the world, and 4% of the deaths also. Despite we have some research in COVID-19, but still very low compared to other countries. Uh, about quarter of this research done in Egypt, and we have in Egypt still we have low scientific background. I mean, the labs is not well equipped like Europe or like USA or China. So we need more funding for research in Africa, in Egypt. And I will show today some work that I have done with my colleagues in Egypt on COVID-19. So we have to talk about targeting strategies against COVID-19. We can target COVID-19 by vaccine or by antivirals or by immunomodulators. Today we have some vaccines that approved by FDA, and sure it will affect the spread of the virus, but we still have we are in need of antivirals and other vaccines that can protect people against COVID-19. So my work today will be at mostly on the vaccine and antivirals against COVID-19. First, we have to a very short background about the virus. As we can see here, there is structural proteins that form the structure of the virus, and there are non-structural proteins that function inside the host cell. So I will focus today on the spike protein, which is the protein that makes the coron-like shape of the virus, and it comes from its name, coronavirus. The spike protein have two conformations, the post-fusion and pre-fusion conformations. The pre-fusion conformation is the most abundant conformation on the viron. There are three different conformation of the pre-fusion: the closed conformation, the one open and two open conformation. This part called is the receptor binding domain of the spike, and this domain should be open in order to interact with the host cell proteins to make the recognition and entry of the virus. So the function of the spike was the host cell recognition. It recognized the host cell. Then it's the entry of the virus into the host cell and escaping the viron from the host immune system. As we can see here, there is a lot of glycosylation sites in the spike protein that make the virus not visible for the immune system. I worked on host cell receptors called glucose regulated protein, or GRB78. GRB78 is a chaperone protein found in every cell in the host in the human. Its function as the master of the unfolded protein response, which is it affected by the stress. When cells are under stress, it will overexpress this protein to alleviate the stress by making the unfolded proteins go through endoplasmic reticulum associated degradation mechanism. The unfolded proteins will be break down or refolded to be in the correct conformation. So GRB78 is important protein in the cell, and its function is very important for cell viability. They found that GRB78 can be localized to the cell membrane of the host cell and called it cell surface GRB78. This cell surface protein can recognize different pathogens like viral pathogen or other pathogen based on fungi. So this protein can help the virus or pathogen to enter the cell. This is reported for Zika virus, for hepatitis C virus, for dengue virus, and for MERS COVID and COVID-19. So GRB can help the coronavirus to enter the host cell. We predict this this is the mechanism of the ER stress. This is a normal condition, and this is under stress. The GRB is overexpressed in the endoplasmic reticulum and escape to the cytosol and then to the membrane when it can recognize pathogenic fungi or viral. So we have modeled the interaction between GRB78 and COVID-19 and published it in in March 2020. We built the model for SARS-CoV-2 spike protein because it wasn't solved yet. And then this is the structure. This is the model that we built for SARS-CoV-2 from SARS-CoV. And we have a very high sequence identity with 77%. So our model was good based on these values. And then we make sequence alignment between the different coronavirus spike proteins and found 4 regions that can recognize GRB78. After some validation based on the sequence identity and the hydrophobicity of these sequences, we can see that region 4, which is this part, is the most suitable region that can recognize GRB78. So this is a region 4. You can see here, this is a black ribbon here. This black region is cyclic, so it resembles the BIB42. BIB42 is a cyclic peptide that used before as an anti-tumor, as GRB78 is overexpressed in some types of tumors. So they try to conjugate the cyclic peptide with chemotherapeutic in order for these chemicals to enter to target the tumor cell, and they were successful for tumors like the not solid tumor, because as you know, the solid tumor, there's some problem with the internalization of these peptides. So I mean, this peptide was used before for cancer treatment. This region of the spike resembles that cyclic peptide. So we can see this part of the spike can act like the cyclic peptides that target GRB on the cell surface. So this is our model that we built between the spike protein and the GRB78. This is the interaction region. You can see here the amino acid residues that take part in the interactions from the spike protein, the red one, and the substrate binding domain beta of GRB78, the cyan one. Mainly, the interaction is hydrophobic because, as you know, BIB42 or GRB78 is a protein that recognizes unfolded proteins. So usually, it recognizes the hydrophobic patch on the protein surface. This is the complete model of the GRB78 over the cell membrane and the spike protein over the coronavirus. And this is the interaction that can help the coronavirus to enter the cell. After that, we have a lot of spike protein solved structures available in the protein data bank. So we repeat this modeling study using the solid structure and found that spike protein can be recognized by ACE2 as the main receptor for coronavirus. This ACE2 is conjugated to the membrane through this protein. So our model shows that the GRB78 should be carried out on some protein on the membrane. I mean, GRB is not inserted into the membrane directly, but some protein can carry this can carry GRB78 in order to be available for spike recognition. So this is another model that we built based on the solid structure of spike protein. This study, we say that the recognition site of GRB78, which is this region from the spike, can be used as fingerprinting for some vaccine. As we can see here, the four immersed human coronaviruses that we took in the first of the lecture, NL63, 229E, OC43, and HKU1, have these fingerprint parameters that are found in the SARS-CoV-2, which is the first one is the cycle is uh, amino acid C and another C that can make disulfide bond, and in between there is a residue that have some different criteria. I mean, in this flexibility. So we have proline in all of the immersed human coronaviruses and G glycine in the SARS-CoV-2. So the cyclic form can be formed easily for this residue in the middle of the chain, of peptide chain. So we can use this, I mean, the immersed human coronaviruses can be used as a vaccine against COVID-19. We say that in the paper, that we can use immersed human coronaviruses that has very mild symptoms of cold-like or flu-like symptoms. So we can use it as a vaccine against COVID-19. Here's the interactions that occurred uh, between spike protein receptor binding domain and GRB78. So we show here the three residues that can be used as a fingerprinting, the C48 and C40, C488, and the glycine, I just don't see the number, but yeah, glycine in the middle of the chain, the blue chains. So we try to find the possible binding site from different viruses like Ebola virus, like human papillomavirus E6, and Zika virus and hepatitis C virus. So we modeled the interaction sites that can be used as a recognition site for GRB78 for that viruses to enter the cell. So now the targeting strategy is based on targeting this cell surface GRB78, which is expressed over the membrane of the stressed cells. So we can inhibit the receptor binding domain, this domain of the GRB78, by some cyclic peptide or monoclonal antibodies or phytochemicals to inhibit the recognition function of GRB78 to the viruses or pathogenic.
Atto, could you start
Yes.
to wrap up your talk, please?
Sorry.
Yeah, your time is over. Could you
Ah.
start to wrap up your talk? I'll give
Okay.
you another
Okay.
2 minutes to wrap up.
Yeah, okay. So I just have 2 minutes. So I can see that we have some natural compound. We try to use it as a binding site to this GRB78 to inhibit it. And finally, we have some work on the new strains of human coronaviruses. We found that the host recognition is enhanced in the UK strain, and this is the wild type, and this is the UK strain of the receptor binding domain. We found that the recognition is enhanced in the new strains. Finally, we can conclude that we can target GRB78 to inhibit viral recognition of this protein and to be able to inhibit the cancer progression and also the viral recognition. So it is two, two birds with a stone, if I can see that. Okay, so I just finished my work. So thank you for all my collaborators from Cairo University, Professor Wael Shimi from University of Alberta, Dr. Khaled Barakat from University of Lund, Professor Salama Karadaghi, and from ICTB, Ali Hassan Ali. And thank you for all of you for this invitation. And I wish we have some work together in the future.
Hey, thank you very much for
Thanks.
a very nice presentation on obviously a very important topic. So the floor is now open for questions, and I'll accept one or maybe two short questions. Yes, Emmanuel, please go ahead.
Yeah, thank you very much for this very impressive topic. I
Thanks.
was just wondering, is it possible that you could do some biochemical assays, binding studies, in particular to validate the binding of the GP, the
Yeah,
GRP
GRB
78
78.
and the spike protein.
Yeah. Okay, so I started some collaboration in Egypt. We don't have funding for that, and I don't have the experimental qualification for this work. But I have started collaboration with people in the UK, and we are now make some adjustment for applying for some funds in UK that we can use some cyclic peptide to against GRB78. And also I have applied for funding here in Egypt, and I get fund from Academy of Scientific Research. So yeah, in near future I will work in some experimental validation for this hypothesis.
Thanks.
Thank you.
Okay, thank you. There's one question from the chat from Isabella Felly.
Yeah.
She asks, are there any other disordered proteins from COVID that could be targeted by GPB78?
Okay, sure. GRB78 is targeting all unfolded proteins. So, I mean, whatever this unfolded protein are from host cell or from the coronavirus, it should be targeted by GRB78. And I think there is a paper in Science say that GRB78 is bound to spike protein, and they suggest that binding is based on the unfolded in the spike protein. So the GRB78 makes some binding due to the spike protein have some unfolded regions. So yeah, sure. If there is some unfolded protein based on the spike, it will be recognized by GRB78 and other chaperones in the cell.
Okay, thank you. Any last pressing question? If not, let's give the speaker another round of applause. Thank you very much for a very interesting and very nice work.
Thank you very much.
Right.
Thanks.
If there are more questions, you can obviously continue in the chat to ask the questions, and they can be dealt with during the next talk. Right. So our next speaker would have been Olamide JJ, and I see that she is around, but she has asked whether her talk could be moved to Wednesday. So I just want to confirm that. Is that still the case? Or would you like to give your talk now?
Sorry, I was thinking I'll do it at the end as the last participant for today.
For today?
Yes.
So not on Wednesday, but today. You want to have the last talk?
I'll do it.
Session.
Yes, I'll have it as the last talk.
Today and not on Wednesday.
Today.
Yes. Okay. Okay.
Okay.
Sure. Let's do that. Right. Okay. So then we'll move the other talks up a little bit. So the next speaker is Eloy Valina, but I also don't see him in the participant list, unless he has logged on with another name. So is he around? Eloy Valina. Okay, if not,
he was here earlier. Actually, he there he
okay.
is. No, I'm sorry, that's a different person. He was here. I just got an email from him.
Oh, really? Okay, maybe you can just tell him that he's welcome to speak as soon as he logs in. But yeah, let's continue with the next speaker, and when he shows up, then he's welcome to give his talk. So there will be a little bit of shuffling the speakers this afternoon. But I see that the next speaker is around. So that is Riyad Mayet. He's from the University of the Witwatersrand, or WITS. And he's going to talk about the characterization and crystallization of the TBR1 T-box domain in the presence and absence of the T-box binding elements. So yeah, you're welcome to share your screen, and then the floor is yours.
Oh, there's Eloy. He's connecting.
Yes, I'm connected. I'm just waiting to share my screen.
Oh, okay. All right. So let me do this.
I see
Let
that
me.
Elo is here now. So
So what do we want to do? We want to do Ilya? You call it, Tart. What do you want to do? What are we
yeah,
doing?
yeah. Let's go according to the schedule. So Eloy, you're the next speaker because the one before you is I'm going to talk a bit later. So yeah, we are
So
looking.
Eloy, you should be able to share your screen now.
Yeah, you.
Right. Yeah. I'm ready.
Okay, good. Yeah. So Eloy is from the University of Stockholm, and yeah, thank you very much for joining this conference. And his talk will be on evolutionary cues in the physicochemical description of proteomes. Right, the floor is yours, Eloy.
Right. Thank you. I guess all of you can hear me probably now.
Yes.
Right. So my name is Eloy Valina, and I'm doing my PhD at the Department of Biochemistry and Biophysics in Stockholm University. And today I'm going to be talking about what is the second project of my PhD, which has to do with this evolutionary cues in the physical chemical description of proteins, which I suppose will be clear. It will be clear what all of this means by the end of the presentation with any luck. Right, so all of this started at the beginning of my PhD by recognizing that the cell is a pretty messy system from the point of view of a physical chemist. It is very crowded. It is up to 40% particles, protein and nucleic acids in volume. And it is made of a wide range of shapes and sizes, which makes for a difficult sort of description of it. And in this mixture also, small molecules participate. And despite all of this, protein mobility and solubility remains very high in the cell, which is kind of surprising compared to the systems that we're used to studying, right? So this is the first striking observation that we recognized. And then in the first project in which I participated, I collaborated with people from the University of Lund into sort of laying the theoretical framework of describing how the cell as a system achieves this solubility and high mobility of the proteins. And this is a fascinating paper. I recommend everyone to read it. But the main take-home messages of this paper were that it is the electrostatic interactions in the cell that ensure this situation. And this is the result of a compromise between two requirements, which is that on the one hand, proteins need to keep away most of the time and stay at a safe distance, so to say, so that they don't become too slowed down by other interactions. But also, the regime should allow close contacts to occur so that specific interactions between proteins do happen, right? And the parameters that we concluded were critical for this regime were particle size that we are always estimating by protein molecular weight, and net charge density, which is a measurement of the amount of electrostatic charge distributed over the surface of the protein as if it were a smooth sphere, so to speak, right? And these are the two parameters that control proteome solubility, something that we've also called colloidal stability in the cell. And during this project, we also realized one more thing, which was that the net charge density distributions of different organisms were not all of them the same, and then there were striking differences from one organism to another. Here's an example, for example, from that paper in which we compare E. coli, our model bacterium, with Halobacterium salinarum, which is this halophilic archaeon I'm going to be talking about a bit more later. And then we recognize that the electrostatic charge density in Halobacterium salinarum can be 10 times as high as the one that you observe in Escherichia coli. And these we connected with the lifestyle of Halobacterium salinarum. So salinarum is a halophilic archaeon that lives in very salty environments. And its strategy to deal with this salty environments is to accumulate electrolytes inside the cell. So it tends to accumulate potassium chloride, right? This creates, from our physicochemical point of view, a high screening environment in which electrostatic interactions are dampened. And so the proteins respond by accumulating more negative charge, and so maintaining this mild electrostatic repulsion that ensures protein mobility and solubility. Right, and it was this recognizing these differences between E. coli and this other organism that sort of made us wonder if we could extend this to any other organism or virtually to all organisms, right? So the question for this project is whether physicochemical properties such as net charge density change across all organisms. And then the basis of this question is that proteome solubility and protein mobility are subject to natural selections in the sense that they affect cellular fitness. They are, of course, inheritable in the sense that they are encoded in the genomes, much like any other feature, and they are tractable in as much as we can formulate proxies for these properties, right? So it makes it an amenable target for following changes across different organisms, and hopefully relating them to evolution. That is what's going to be the aim of the project. The way in which we chose to address this question was by formulating this. So starting with these two properties, net charge density and molecular weight, we've calculated these for all of the proteins in these proteomes, and then the proteomes you can simply get from NCBI. These are like long sequences from which you find like all of the proteins encoded for a certain organism, right? And then rather than focusing on the nuances of the distributions, we simplify this down to the average property value, right? And then if we do this for two proteins, then we can come up with these two-dimensional coordinates that we can extend to organisms in bacteria, in archaea, but also in eukaryotes. And even though this requires some computational effort and especially some memory for dealing with where to store like all of the sequences, as a computer project, it's very minimal in the sense that these properties are calculated very simplistically, only from the sequences, and reducing them to the averages also makes the coordinate maps very minimalistic. So it is amenable to anyone with sort of any kind of access to computer. And in fact, I did all of this project on my laptop, basically. And by extending this mapping of physicochemical coordinates to a two-dimensional space, then we created this first sort of physicochemical map of life, right? And then here we're looking at more than 30,000 bacteria, more than 2,000 archaea, and more than 2,000 eukaryotes. And the panels here on the right are supposed to help you gauge the amount of observations in each of these squares. So there's truly a lot of information here. And already from this overlook of the life diversity in the sort of physical chemistry, we saw two striking things. One of them was that eukaryotes are mostly spread on the vertical axis, and they are very narrowly confined to certain values of net charge density. And on the other hand, prokaryotes are widely spread over the x-axis, meaning that they change a lot in net charge density, but not so much in molecular weight, which is something that we didn't anticipate at all, and is just sort of fantastically captured by this very simplistic plot. The other thing that we recognized already at this point was that there were some groups of interest, some of these observations constitute outliers within the group. So within bacteria, archaea, and eukaryotes, we find this sort of deviating observations, and we wanted to figure out why these organisms were so divergent, right? And so I started looking at this like groups of features, right? And the first one that I came around with was an old friend, which was this group of interest in archaea. So the archaeal cell can be divided into these three clusters, pretty trivially actually. And then the cluster one captures all of the halophilic archaea, like that Halobacterium solinarum I mentioned before. And then again, this is because all of them sort of share this adaptation to salt-rich environments like this evaporation ponds in sunny places like the Canary Islands, not so much in Sweden, unfortunately. And because they share this salting in strategy, then they also share this adaptation to their own cellular environment by accumulating more net charge density on their proteins, and in turn maintaining this mild repulsion between them to ensure solubility and mobility. So this was like a first positive result that was quite encouraging. The other two clusters are a bit more difficult to map, but the cluster 2 turned out to be 60% unclassified archaea, which is quite surprising. These are slightly high in protein size, and then 25%, the ones that are classified, 25% of those are belong to this phylum Candidatus Posidonia, which are archaea typically found in the sea surfaces. So one immediately would like to connect these two unclassified archaea with the ones that are classified, right? And by argument of similarity, say, well, maybe this unclassified archaea will belong to this family as well, right? I don't want to like jump the gun just yet. The other larger group is a bit of a catch-all box. This is where you find all of the other phyla of archaea, and in particular, all of the newer phyla that are not your traditional airy Archaeota, but like the newer ones that have been discovered in the last couple of decades. All of them belong in the third cluster. So this already gives us a sense of the sort of classification power that these physicochemical descriptors might have. In bacteria, rather than a cluster, really, what we look at is this tail of very positive proteomes. So these are organisms whose proteomes are on average very positively charged. And this was also surprising because I said before, electrostatic interaction should be repulsive, and this truly could be achieved with positive and negative charges. But we expected negative charges because the nucleic acids are, of course, negatively charged as well. So it only makes sense that proteins would remain soluble and colloidal stability would be maintained on that regime of negative charge. So when we looked at these organisms more closely, what we found was that these were typically small proteomes, the likes of which you would find in insect endosymbionts, and these are bacteria that live within bacteria that in turn live within these insects like the cicada and the aphids, which are like quite remarkable organisms, and very minimal proteins that are enriched in nucleic acid binding proteins. So our argument at this point is that these organisms might have gone through a process of purifying selection, a bit similar to what some people believe has happened to mitochondria, right? And how like their proteomes have progressively lost certain functions and delegated those into the host cell. And then finally, for eukaryota, this is the group of interest that we could find, and then it's maybe not very clearly seen here, but there's like a small cluster of observations around these coordinates. The borders don't matter too much, or I don't believe they matter too much. But then within this particular bubble, what we find is exclusively parasitic eukaryotes, and in particular the ones that are responsible for causing deadly diseases like malaria, for example. And then many of the species related to different types of malaria are found in this cluster. And this is a bit of a bittersweet moment in the project, because even though we have this very clear observation, we have no compelling explanation of why this would be the case. There's no trivial reason why malaria proteins would be longer or bigger, if you will, or would have more net charge density, more positive net charge density than other eukaryotes, or at least we haven't really found any reason why this is the case. If anyone has any ideas, I'd be very keen on listening to them. But this is what I'm trying to underline here, that we don't always have explanations for this. Okay, so all of that came only from looking at molecular weight and net charge density properties in a two-dimensional space, but we do know a bit more about organisms, right? And I've mentioned already the taxonomical hierarchy, I think. So the established classification of organisms is already there in a resource that we can easily access and correlate to our proteomes. And there are other physical-chemical properties that we can look at, and even we could try to find other observables, right? So I'm going to show you like a few examples of this without making this excessively long. So coming back to bacteria, for example, here are the major phyla of bacteria, and again, here is that positively charged tail that I mentioned before. But even within the bigger sort of blob of observations, we can see that there is a systematic ladder of these groupings of phyla, right? Like, not every phyla is sort of evenly spread over this map of coordinates, which is quite striking. And then if we then turn these observations and move them to a new two-dimensional space that we define by two other physical-chemical properties that are also easily obtained from the sequences. And in this case, I'm looking at the GC content in percentage of these genes, and also like their charge residue fraction. So rather than looking at the net charge, net electrostatic charge, I just look at charged residues of whatever sign on the surfaces of these proteins. We also recognize some of these tendencies. Some of them are familiar to us from basic biology courses, right? And then we have actinobacteria, for example, the typical high GC gram-positive bacteria, and the low GC gram-positive bacteria also unsurprisingly found more on the left side of this axis. But we see some groupings also for Tenericus and Bacteroidetes that are quite interesting. And again, I can't really give you a reason why this is the case. I'm so like reporting the basic observation that this does happen, and this is what at this moment the project that I'm working on is based on. And then I promise you an extra observable, and then this is really just gram staining. So gram staining is a bit of an ill-defined concept nowadays, and it's not very useful for proper phylogeny, but you can still sort of you still find it annotated in databases, and it's useful for dividing organisms, as we've seen. And then here, for example, if you divide all of the bacteria into gram-positive or gram-negative, if they have like such annotations, then you find that there's a very clear bias also. So no big in size, but fairly systematic towards gram-positive organisms having more negative net charge density distributions. And again, there's no, I can't give you any complete reason why this is the case. In the paper that we're writing right now, we discuss this a little bit in detail and what this, how this might be related to the properties of the membrane of these organisms. But this is like the basic observation that I wanted you to see today. The take-home messages from these projects that I'm hoping you remember is that we can use physical-chemical parameters as descriptors of taxonomical variation. And they are much more simplistic than phylogenetic, proper phylogenetic analysis. They don't replace it, but they might be complementary to proper phylogeny, right? In some cases, these physicochemical descriptors can be related to niche and lifestyle of different organisms, which is very cool. And in others, we just observe the tendencies, but we can't really give you explanations just yet. And that's the explaining part of the project is what we're working on right now. And then we would argue also that in difficult cases of phylogenetic classification, or as a way to clean up currently classified organisms, one could consider this sort of physicochemical matching and physicochemical similarity in terms of the simplistic dimensional spaces as an argument for introducing a new organism or removing a new organism from a certain taxonomical box. So that's all I wanted to say. I like to also take a minute to recognize the work that I've been doing at the Department of Biophysics and Biochemistry for all of their support, and in particular that of my supervisor, Michael Oliveberry, and all the people in the group. And also as for the first part of the presentation and my first project in my PhD, I work with Håkan Wennerström, which is at Lund University. So thanks everyone for listening, and I'll be happy to take any questions.
Yes, thank you very much for a very interesting talk. So the floor is now open for questions. Emmanuel, do you have a question? I see your mic is on.
This is a very nice presentation. Thank you very much. I was just wondering if there is any correlation between the physical chemical property and the stability of the of the different proteins. For instance, I know that the bacteria proteins are more stable than the eukaryotic ones. But I was just wondering if you see any correlation in that sense.
It's difficult to compare, say, as you said, bacterial proteins with eukaryotic proteins in base of these very simplistic physicochemical descriptors. But something that I could say is that these properties are related to stability inside the cell. And then we know, for example, that proteins that are better capable of maintaining solubility inside the cell are also more stable, in the sense that the cell affects protein stability negatively, but proteins, through the properties of their surfaces, adapt to that sort of negative impact and manage to remain stable inside the cell. So it's certainly related, but I don't know how eukaryotic proteins and bacterial proteins might compare.
Okay, thank you. Any last pressing question? It's time for a very short question. Okay, if not, let's thank the speaker again for a very interesting talk. And if you have more questions, you're welcome to write them out in the chat. And we can continue the discussion during the next talk. So thank you very much.
Thank you.
Right, we're slightly ahead of the schedule. So we will continue with the next speaker, which is Riyad Mayet from the University of the Witwatersrand. And he's going to talk about the characterization and crystallization of the TBR1 T-box domain in the presence and absence of the T-box binding elements. Right, Riyad, you can share your screen and then the floor is yours.
Hello, everyone. Can you see my screen?
Yes, we can see it. You're welcome to continue.
Okay. Good day to everyone. My name is Riyad Mayet, and my project deals with the characterization and crystallization of the TBR1 T-box domain in the presence and absence of the T-box binding element. Okay, so a little bit of an introduction. The TBR1 T-box is a transcription factor. It is a neuron-specific transcription factor that is involved in brain development, and more particularly cortical development. It is involved in differentiating neurons into their respective cell types within various layers of the developing neocortex. The TBR1 T-box transcription factor belongs to the T-box family of transcription factors, which share an evolutionarily conserved DNA binding domain known as the T-box. And we'll come look at the structure more in detail a bit later. So the DNA binding domain is monomeric under reducing conditions. And what makes this protein interesting is that it has recently emerged as a master regulator of genes implicated in autism spectrum disorders. And variants of this protein have also been shown to cause sporadic autism in various cases. Another thing that's interesting about the protein is that it interacts with the FOXP2 transcription factor, which is the first gene product that has been associated with human speech and language. So aberrant molecular interactions between TBR1 and proteins like FOXP2 could explain why there are speech disorders in neurodevelopmental disorders like autism. There's no crystal structure for the TBR1 T-box, and the DNA binding mechanism is unknown. So as I said earlier, the TBR1 T-box belongs to the T-box family of transcription factors, and they are characterized by the T-box DNA binding domain as shown over here. So this is the T-box domain from another T-box family member, TBX1, and the domain consists of 180 approximately amino acids and weighs about 20 kilodaltons. So this domain is both necessary and sufficient for sequence-specific DNA binding. So all T-box proteins, because they all share this T-box domain, they all bind an 8-base pair consensus sequence known as the T-box binding element. And there is some transcriptional activity in the C-terminal region within the T-box DNA binding domain. And now I just like us to zoom a bit into the structure. And if you look at the top right-hand side of the structure over here in yellow, we see the characteristic fold of T-box proteins. It is called the immunoglobulin-like fold. So what it consists of is the 7-stranded beta barrel, which is enclosed off by 2 antiparallel beta sheets. And then also it has these 2 mutually perpendicular helices, which lock the protein into a fixed state and wedge it into the minor groove of the DNA. Now, the T-box structures have not been solved in the presence and the absence of the T-box binding element for most T-box structures. Only in one T-box structure do we have it in the presence and the absence of DNA, and we can only get the DNA binding mechanism from that structure. Okay, so the issue is that even though these proteins have an evolutionarily conserved DNA binding domain, the fact that they have different expression patterns, for example, this protein TBR1 is expressed in the brain, whereas the one with the solved crystal structure TBX5 is a cardiac specific. So perhaps they have different expression patterns as well as different neuromolecular networks. So the aim of this study was to characterize and crystallize, in particular, the human TBR1 T-box transcription factor in the presence and absence of the T-box binding element. And the reason why we wanted to do this was to determine the mechanism and kinetic parameters of DNA binding, as well as to provide more insight into the molecular function of the protein, and maybe try and understand how the intermolecular interactions with other proteins in DNA affect neurodevelopmental disorders and gene expression. So in a broader sense, this could enhance our understanding of how gene regulatory networks will affect brain development in neurodevelopmental disorders like autism. So this is a summary slide of my methods and my results. So we'll begin with overexpression and purification. So in order to obtain sufficient quantities of the protein for downstream characterization and crystallization, it was overexpressed in E. coli cells by use of an expression vector. So basically the gene sequence for the human protein is expressed in E. coli and then purified. So with regards to purification, the protein obviously needs to be purified for it to be characterized, because we want a homogeneous solution for setting up crystals. And the first step to do that, we used immobilized metal ion affinity chromatography. So in this type of chromatography, the column is lined with a Sapporo solid support matrix, which is covalently bonded to a chelating agent, such as aminodiacetic acid. What this does is it chelates a metal ion, as you can see over here. And then the protein has a hexahistidine tag encoded into it. So it makes dative covalent bonds with the metal that has been chelated by the chelating agent. So now we have protein bound to the column, and in order to elute protein for the column, a competitive inhibitor such as imidazole will be added to displace the protein. And so now we have mostly pure protein. So this is what the elution profile looks like. So the flow through represents everything that did not bind to the column. The salt wash represents a rinse of the column where non-specifically bound proteins maybe could have come off in this peak. And the last peak over here is the elution peak, which we are interested in as it contains the pure protein. Now, in case the protein hadn't been 100% pure, or not really 100%, but really pure, close to 100%, size exclusion chromatography was applied. Size exclusion chromatography is also known as gel filtration chromatography, and it separates molecules based on their molecular size. So if you look at this diagram over here, this represents a column, and the blue represents porous beads in the column. So if you pass a mixture of molecules, large in red and small molecules in pink, through this column, the larger molecules will not enter the porous beads, and so they will elute first, as you can see on the chromatogram. But if the particles are smaller, they will move through the porous beads and will elute in an order of biggest to smallest, since the smallest particles will have the longest space to traverse. So they will elute later. So here is the elution profile from my size exclusion chromatography. And what it shows that under reducing conditions, the TBR1 T-box transcription factor is predominantly monomeric with a size of 26.2 kilodaltons. And there is, even in the presence of a reducing agent like DTT at 2 millimolar, there's still the formation of a dimer, even though it's at low concentrations. But more importantly, this enabled us to separate out the monomer from the dimer so that the monomer could be used for crystal trials, since we want a homogeneous solution. So just to better understand how the whole overexpression and purification worked, and to compare the results from various stages, polyacrylamide gel electrophoresis was used. And in this method, proteins are linearized, coated with a negative charge, and then put into a porous gel. And under an applied voltage, they will migrate through the gel. The bigger proteins will be trapped in the gel, so they will appear as bands at the higher up, and smaller proteins will travel through the gel easier and will end up towards the bottom of the gel. There's also a set of size standards over here. So in the overexpression stage, where we added IPTG to induce overexpression of the protein in bacteria, we see that the protein, since it's around 25 kilodaltons, we should find it in this region over here. And we can see that there is expression in the supernatant as well as the pellet. So we decided to proceed with the supernatant since it will be easier to work with soluble protein, and there was a high enough amount for us to work with. So this is the sample essentially before purification. Then after the immobilized metal ion affinity chromatography, this is the result. So we can see that most of these proteins have been purified. However, there are still some contaminating bands that maybe have resulted from non-specific binding. And so to remove those bands, that is why we employed size exclusion chromatography. And under reducing conditions, the dimer was shown to be 2 reduced monomers, each of 25 kilodaltons. But more importantly is the last column over here, where we can compare the final protein that will be used for characterization and crystallization. So if we compare from here to here, we can see that all the contaminants have been removed, and pure protein, which is just greater than 25 kilodaltons, can be used for the downstream applications. The purification and overexpression protocols obviously were optimized in our laboratory. So once we now have protein in sufficient quantities and protein that is pure, we can begin with the structural characterization of the protein. So an easy way to characterize the tertiary structure is with intrinsic tryptophan fluorescence. So just some background on the technique. Proteins have intrinsic fluorescence as a result of aromatic amino acids present in the amino acid chain. Most of this fluorescence is due to the indole ring present in tryptophan residues. And when tryptophan is excited at 295 nanometers, it will emit light at a wavelength which depends on the exposure of the tryptophan residues to the solvent. So when proteins are folded, typically because tryptophan residues are hydrophobic, they tend to be buried within the core of the protein. Whereas in unfolded proteins, these tryptophan residues would be exposed to the solvent. And we can measure the exposure to solvent by looking at where the emission maximum wavelength is. So if the tryptophans are exposed to solvent, it causes a redshift. Um, so here is the results of my intrinsic tryptophan fluorescence. So what we used with, or what we done with this technique was, we first got a spectrum of the intrinsic tryptophan fluorescence of the protein in its native folded state, or what we think is the native folded state. And then we get another spectrum of the protein that we know has been denatured, or that we denature using guanidinium HCl. And you can see that from here, the maximum wavelength is about 338. And here, the maximum wavelength is about 348. So this shift from the pink to the green spectra show that the protein was initially folded. Because if it was not folded initially, the spectrum would have looked like the green spectrum. So from this, we can confirm that the protein is correctly folded. Okay, so once a protein had been characterized structurally, we then proceeded on with functional characterization. So an electrophoretic mobility shift assay is used to determine or detect whether a protein binds DNA. And the reason why we would do this is to confirm that the TBR1 T-box transcription factor is indeed binding the T-box binding element as it should, so that we can then use it in crystal trials. So typically the first lane is a control lane, which contains just free DNA that's labeled. And it migrates relatively easy through a porous gel because it is relatively small. If you look at the second lane, when protein is added to the labeled oligonucleotide or DNA, two bands will appear if the protein binds some of the DNA. So this band here would correspond to unbound DNA, and this band would correspond to protein-bound DNA. And the reason why it's shifted in that direction is because the formation of a complex will result in a larger molecule, which will then be retarded more in the gel, causing a shift. And if you look at the third lane, this would be typical of when the protein would bind all the DNA present in the sample, and there would only be one band at the higher complex. So here are the results of my electrophoretic mobility shift assay. And what it shows, if we look just at the first lane, this is the control lane, which has just the labeled T-box binding element. And then from lanes 2 onwards, we titrated increasing concentration of the TBR1 T-box domain. And what it shows that even at low protein concentrations, the TBR1 T-box domain still binds specifically to the T-box binding element. We can see from this lane over here, so at higher concentrations of about 5 micromolar, that all the protein is bound. And we can even see the formation of some higher order complexes, even though a reducing agent has been added. And this is interesting because the higher order complexes are dimers that are disulfide linked. So that may be worth looking into a bit more. So this confirmed that the protein was functional and able to bind DNA in the way that it should. So in order to get a better understanding of the DNA binding of the protein, we employed fluorescence and isotropy to determine the dissociation constant. So in fluorescence and isotropy, a polarized excitation light is used to excite your fluorescent molecule. Now, if the fluorescent molecule is very small, it will rotate rapidly in the solution. And this will cause the polarized light to become depolarized. So the emitted light will be depolarized, resulting in a low fluorescence intensity or low fluorescence polarization signal. However, if the molecule is larger, for example, a fluorescent DNA bound to a protein, the rate of rotation in solution will be much slower. And this will result in the emitted light being more polarized or less depolarized, which means that the fluorescent polarization or fluorescent intensity signal will be higher. So this technique can be used to study saturation binding and to obtain binding curves. And in specific, we have used a one site specific DNA binding model to show that the KD is approximately in the nanomolar range, which means that there is relatively strong binding. And the reason why we would also do this is to get an idea of what concentrations to use in crystallography, as well as to provide a baseline for which or against which autism-causing mutants can be compared.
Riyad, can you start to wrap up your talk, please?
Yes, I have two more slides. So once a protein had been successfully characterized, both structurally and functionally, I then attempted to set up crystal trials in the presence and absence of DNA. However, so far, I've only set up crystal trials in the absence of the T-box binding element using vapor diffusion and microbatch methods from low to high concentrations. However, these trials have all been unsuccessful, and we think that the reason is the instability of the protein in the apo form. So we believe crystal trials will work better, or hopefully will obtain protein crystals when we crystallize in the presence of a high concentration of the T-box binding element. I just want to acknowledge the National Research Foundation for funding the project, my supervisor, Dr. Sylvia Fanuki, for support, and everyone at the PSFRU for their assistance. Thank you.
Right, thank you very much, Riyad, for a very interesting talk. So the floor is again open for questions.
Is there one in the chat?
Lawrence, do you have a question?
No, I was just looking to see if there's one in the chat. Is Patrick, is that a question or an applause? I guess that was an applause.
No, it's an applause. It's an applause.
It's a great talk.
Yes, Emmanuel.
Yeah, I just want to make a couple of remarks. I really like your presentation. I really like the purity of your protein is like 100% pure. Yeah, I totally agree with you that if you try to crystallize without the apo form, that might be unstable to crystallize, and definitely in the presence of something to keep it stable is the best way to go. So you could also try other crystallization methods, not vapor diffusion, not only vapor diffusion.
Thank you for that.
No worries.
Yes, there's time for another talk, another question, a short one maybe. Okay, if not, let's thank the speaker again. Thank you for an illuminating talk. And if there are more questions, then obviously you can continue to discuss in the chat. So thank you very much. Right, the next speaker is Kenechi Kanayo Onyia, if I pronounce that correctly, from the University of Nigeria, who's going to talk about fluorinated surfactants for membrane protein extraction. Right, you're welcome to share your screen. And then the floor is yours, Kenichi.
Okay, thank you.
Hold on 1 second. Actually, I do not. What's oh, here we are. Okay. Yep. Okay, so I think you can share your screen now. Can you hear me? Are we still on?
Yeah, I can hear you.
Okay.
Can you see my screen?
I cannot yet.
Can you see it now?
No, let me see. Is there a setting I did not do? I think you should be able to share now. Let's see.
Okay.
Yep, you're a co-host.
Okay. Okay. So
There we go.
yeah. I don't know what's happening here. Excuse me. Can you see my screen?
Yes.
Yes.
Wow. So first of all, I would like to introduce myself. So my name is Kenichi Kanayo Onya. So I'm from Nigeria and I'm doing a PhD, a binationally supervised PhD from University of Nigeria and University of Kaiserslautern, Germany. So I'll be presenting part of my PhD project, which is fluorinated surfactants for membrane protein extraction. So what we are interested in is the integral membrane protein, which is embedded in the cell membrane, because this integral membrane protein is very difficult to study in vitro because of its hydrophobic nature. Therefore, substances like amphipathic substances that contains polar and non-polar parts are used to study this particular type of protein. Detergent is an example of such compound that can be used to study integral membrane protein. But the problem is that detergent has one tail, hydrophobic tail, while the typical membrane has two hydrophobic tails. Therefore, it gives you a cone-like shape that is not a replica of the lipid vesicle. And also, the impact of detergent on the cell membrane is actually quite harsh for some proteins. And using the detergent to solubilize some proteins now makes it very difficult because the functionality of the protein of interest is not really determined at the end. Therefore, we want to look out for some other compounds that can do the work of detergent, can also solubilize lipid membrane and extract this protein of interest, which is the integral membrane protein. So this is just like a workflow of detergent mediated workflow for membrane protein solubilization. Here we have the cell membrane containing the integral membrane protein, and we have an amphipathic molecule. Example is a detergent that forms a micelle that will now integrate into this cell membrane and extract the protein of interest. But as I say, there are limitations of detergents. They are poor replica of the system, the lipid vesicle, and also they form aggregates below the critical micellar concentration. And as such, they denature the protein, and it cannot be used for further downstream experiments. So a particular interest to us is fluorinated surfactants, which is also almost the same as detergents, but the only thing is that the hydrophobic tail, it contains fluorine instead of hydrogen. Why we are interested in this particular compound is that the fluorine, which is more bulkier than hydrogen, is hypothesized that it will not interact with the hydrogen of the hydrophobic chain of the protein because they are not interacting. So replacing hydrogen with fluorine makes the amphipathic compound more bulky, then it has more rigid, the flexibility will be reduced, therefore the impact on the membrane will be reduced. It will have like a gentle impact. That's the hypothesis behind it. Fortunately, already there is a publication on it, or commercially available fluorinated surfactant was used to solubilize membrane protein. But unfortunately, only these two compounds are available commercially. So there is need to synthesize new compounds based on the results that have been published for this particular compound. So first we have to tailor these synthesize new fluorinated surfactants based on the surfactant that is available commercially by tailoring the hydrophobic tail and the hydrophilic polar part. So what we did is that the first part is maltase based and the second part is lactose based. So we just tailored it to see which one will give us the most promising property of interest. So the first is determination, CMC determination, because in solubilizing of membrane, in order to solubilize the embedded integral membrane protein, you need to have a compound that will form a micelle, because it's the only micelle that is quite hydrophobic that will extract it. So first we have to determine the critical micellar concentration of this compound that we have synthesized. So we using ITC, isothermal calorimetry method, so that we can determine the CMC of this compound. So first, we determine the CMC of the compound, which is in the millimolar range. And this is quite a kind of thermogram that we see and the results that we see in it. So after determination of the CMC, the next part is to check the morphology, type of micelle that is formed. Is it rod-like or is it spherical micelle that are formed? So we use dynamic light scattering experiment to determine the type of micelle that is formed. And this is just the size, distribution, and the different concentration of the surfactant. This is an example of one of the surfactants that we are working with. As you can see, the size of the micelle increased. The sizes are increasing as the concentration of the surfactants are being increased. This looks like a rod-like micelle that is formed, because if it is spherical, immediately when micelle is formed, it begins to be constant. But because the size of the micelle is increasing, we hypothesize that it will be rod-like. But of course, we need cryo-EM to confirm the shape of the micelle that is formed. Having determined that, the next thing is to check if these surfactants that we have synthesized can actually replace detergents in solubilizing the membrane, and of course solubilizing the membrane protein that we are interested in. First, since we need to show a replica of the membrane, so we need an artificial lipid vesicle. So the lipid that we use is POPC, and we extruded it from the multilamellar vesicle to get a size of about 100 nanometer to 120 to give us large lamellar vesicles That is the range of size that we want to work with. And at that, we introduce the surfactants to the POPC vesicle that is formed. So these are the different surfactants that we worked with. This is fluorinated surfactant, this is cymers, this one is lactose-based, maltose-based. So we want to see the kinetics of solubilization of the artificial lipid membrane. And it is determined by the intensity of light scattering intensity. As light scattering intensity decreases, it shows you that solubilization is occurring. But initially we have POPC that is in the size of 100 to 130, and introduce a surfactant, various surfactants, we begin to see the reaction that the size is decreased to the micellar size of around 10 to 15 nanometers for this particular compound at a particular temperature. And we did it for maltose-based, lactose-based, and for hydrogenated compound. We want to compare the results and want to screen and bring out the compounds that give us the best result. Looking at this, we saw that the maltose-based and the cymers gave the best result because after almost four hours or 2 hours, the solubilization process has already finished. But for the lactose-based, kind of tricky. You needed almost like 16 hours, even sometimes at 16 hours, it has not completely solubilized the lipid vesicle. Based on that, we discarded the lactose base because we are interested in the most promising surfactants, and we went further to further experiment with the maltose base and the cymers and DDM. So the next thing is for us to actually study what is happening in the solubilization process. Initially, we have something like this. This is a typical representation of what is happening, and it's shown in what is called phase diagram. So you want to see the phase transitions from the bilayer. This is the bilayer, the artificial POPC bilayer that we have formed that is in the range of 120 to 130 nanometer. And these are monomers of the surfactants coming in because initially they are monomers, and it gets to a point that it starts forming micelles. And at this point, there is also free surfactant monomers that are still existing. At this point, it starts perturbing the lipid vesicle and starts to penetrate into the membrane. That's the solubilization process. And it gets to another point that is called the solubilization. That's complete solubilization. At this point, you see that the vesicle has totally collapsed, and what you see is only micelle, that is a mixture of surfactant lipid micelle and the free surfactant monomers that are existing. With this, you can plot, represent it graphically in what is known as linear regression graph, where you have the bilayer phase, and we have like a coexistence stage where we have the bilayer plus the micelle. And now we have the complete solubilization stage where we have only the micelle and the free surfactant monomers. From here, you can calculate your data, your saturation point. You can also calculate your transfer free energies. You can also calculate your partition coefficient. So this is just like using DLS, dynamic light scattering, to study it. We have different concentrations of the POPC, different concentration of the lipid membrane. Then we now introduce the surfactant molecules. As you can see, as the concentration of the lipid membrane is increased, then reaching the solubilization point also broadens, it lengthens, showing you that as you're increasing the concentration of the POPC lipid vesicle, also the concentration of the surfactant needed to solubilize it will be increased. This is like a representation of at 0.1 millimolar, 0.3 millimolar, and 0.6 millimolar of POPC. then introduce the surfactant, and have something like this. And we presented it in this diagram. So here we have the concentration of the surfactants on the y-axis and the different concentration of the lipid on the x-axis. The intercept being the concentration of the surfactant monomers, and the slope at this point is the minimum surfactant lipid ratio required for onset of solubilization. Then when it passes through the coexistence stage, you have the minimum concentration of the surfactant lipid that is required for complete solubilization to occur. For us to authenticate this, we needed to study with another method. So also we use ITC experiment to study it. Here, the titrant, you have the POPC in the cell. We have our working buffer in the reference cell, and we have our surfactant in the syringe. And we titrate the surfactants in the syringe into the cell, which is containing our artificial membrane, which is our POPC, at different concentration, because we want to construct a phase diagram. This is a typical thermogram of what we see, and you see also what we observed in the DLS experiment is observed here, that the concentration of the POPC artificial membrane increases, then the width broadens. The width broadens. This is at 4 millimolar 2 and 1 millimolar concentration of the POPC. So we compare the results using 3 techniques. That's DLS, ITC, and NMR. Here, we observe that the results are comparable, as we can see, that the saturation point and the solubilization points at this point are comparable. So from this, we authenticate that the solubilization process of this particular surfactant occurred in a 3-mode phase transitions. Next, we want to study how does this surfactant partition into these membranes, because it's quite important to us to know if they translocate across the outer into the inner leaflet of the membrane, because the membrane contains 2 leaflets, the outer and the inner membrane. First, we use the uptake and release experiments using ITC. Particularly, we are using ITC experiment because it calculates the enthalpy of the reaction directly. So from there, we can calculate other thermodynamic parameters. So from there, here, we calculated the lipid susceptibility factor and the partition coefficient and the enthalpy of reaction of some of these surfactant molecules. We found out that what we see is partial translocation, that the translocation of these surfactants did not integrate into the inner leaflet of the membrane, probably because of non-ideal mixing of the compound, because it is done manually. It is not mixed. It might not be mixed properly. That is why we are getting values that are less than 0.5.
Nechi, could you start to wrap up your talk, please?
Okay, the final thing is to check if our surfactant can extract the membrane protein, which is our interest. So we did an SDS page. So these are the various surfactants, promising surfactants. We compared it with the gold standard DDN. This is gold standard DDN, and these are surfactants that have various concentration. This concentration is their micellar concentration. And we observe that as fluorination increases, that efficiency of solubilization also decreases. But for these particular 2 compounds, they showed solubilization that is comparable to that of DDM. And we presented this graphically for us to know the protein yield of this surfactant, how much of protein we are they able to solubilize This is for other lactose-based. As you can see, it's not looking so good as the maltose-based surfactants. So from this, we can say that we have identified new compounds that can solubilize membrane protein in a manner that is reminiscent to detergents, but with a little perturbation to the order of the membrane. Thank you. So I want to acknowledge my sponsors, that gave me the grant to do the research, University of Kasaslaten and University of Nigeria and Nsukka, that are supervising the PhD. Thank you for listening.
Thank you very much for an interesting talk, Kenichi. So the floor is open for questions. Elvis, does that thumbs up mean that you wanted to ask a question?
Yeah.
Okay, sure.
I didn't know how to do it on my computer. Anyway, so that was a very good talk. I have a very quick question. So were you able to determine the phase transition temperature of the membrane?
No, we did not go that particular way for this. Yeah, we did not go that direction. Yeah, for this particular project. Yeah.
How about the membrane thickness? Because to be able to maintain the functionality of the membrane protein, the thickness has to match with the hydrophobic portion of the protein. Were you able to determine the membrane thickness?
Yeah, that's one of our attributes, because it's still an ongoing project. So we want to use like DSC to determine this membrane thickness and their phase transition temperature. Yeah, we are working on that, but at this point, we have not determined it.
Okay, thank you.
Thank you, Elvis. Emmanuel, your hand is up. Emmanuel, you're welcome to ask a question. You are muted.
Yeah, sorry. Thank you very much for this presentation. It was really awesome. I just have one question in terms of the applicability of the new detergent which you have tried to develop. Because of the fluorine, would you consider to try to use it for phasing in terms of determining the structures of proteins? Have you explored in this sense?
In terms of determining the structure of...
to solve the phase problem for protein structural determination, because the fluorine, you use the fluorine.
Yeah, because I mean, this is still what we would do. As I said, this is just like we are trying it out because we are trying to see if this can replace the detergents, as I said. For this phasing, the downstream experiment, we have not gotten to that, but we will try. Of course, we will try that. Yeah.
Okay, then my last question is in terms of the micelle size.
Okay.
Was the micelle size for this new detergent, was it shorter or greater than DDM?
It was actually, for some, it was quite shorter than DDM. Then for some, it's like DDM, the rod shape, we actually determine very small micelle shape for some compounds. So it differs. That is why we want to tailor, we are like tailoring this surfactant that will give us the best promising properties. Yeah.
All right. Sure. Because I'm looking in terms of structural determination as well. The shorter the micelle size, the better.
Of course, yeah. So the target is to get the ones that have the shorter micelle size, which is quite difficult. Out of the we have screened like 10 compounds, and we were able to just get 1 out of 10 that is like giving us a small micelle. Yeah. So it's quite a difficult task.
Thank you.
Thank you, Emmanuel. Maybe a short question to follow on Emmanuel's question. So how certain are you that there might not be even minor structural changes in when you're using this new surfactant? Because when you use DDM, it is known that that is a poor representative of the membrane, but is your surfactant, the new detergents, much better in that respect to retain all the properties of the protein?
Like in terms of the stability of the protein.
Not only stability, but really the structure, the full structure.
Yeah, because if the structure is retained, that means the functionality is retained, is it not? Yeah. Yeah, so we have tried for two proteins, like we have tried for UMPLA, outer membrane phospholipase A, in the enzymatic activity, and it showed that it performs better than DDM for one of our compounds. Yeah. So some of these compounds, in some aspects, they perform better, but you don't have like one efficient compound that will do all and have the best property for all, better than DDM. So in some aspects, they perform better than DDM, and in some aspects, DDM performs better than them. So that's what we are saying. Yeah. Yeah.
Okay, that's very interesting. Yeah, and thank you very much for your talk. Let's give the speaker a round of applause again. Right. So then the last speaker for this session, we have moved her up to the end, and that is Olamide Jeje from the University of the Witwatersrand. And she's going to talk about a systematic integration of empirical and computational studies to biophysically describe recombinant nicotinate mononucleotide adenyl transferase from Klebsiella pneumonia. That's quite a mouthful. So the floor is yours, Olamide.
Thank you very much. So I'll just try sharing my screen. This is the yes. Okay, so please can you see my screen?
Yes.
Okay, thank you very much. All right, so my name is Olamide Jeje. I'm just concluding my master's degree at University of Witwatersrand, precisely biochemistry department in the School of Molecular and Cell Biology. The title of my dissertation is, sorry about that, a systematic integration of empirical, oh, sorry, how do I take this out? Okay. All right, that's fine. Okay. Sorry about that, please. The systematic integration of empirical and computational studies to biophysically describe recombinant nicotinate mononucleotide adenine transferase from Klebsiella pneumonia and Enterococcus faecium. This research is actually inspired by the spike in the increase of nosocomial infection, which is caused by multi-drug resistant bacteria across the globe, and its impact on public healthcare system in developing countries, especially in sub-Saharan Africa. We see that nosocomial infection is actually an hospital-acquired infection that is contracted during hospitalization, and it includes infections that are diagnosed within 48 hours after hospitalization, 72 hours of discharge, and 30 days. Oh, sorry, what's wrong with this? Sorry about that, please. All right, so I'll just take it all over. Nosocomial infection is actually is also referred to as hospital-acquired infection. So it involves infections that are contracted during hospitalization or prolonged stay in hospital. It includes infections diagnosed within 48 hours after hospitalization, 72 hours of discharge, and or 30 days post-operation. And Klebsiella pneumonia has been highlighted as one of the leading causes of this infection. And it has continued to gain increasing recognition because of its acquired multi-drug resistance strain, which has led to ineffectiveness of most antibiotics, including a class of antibiotics, including the beta-lactams, the aminoglycoside, fluoroquinone classes of antibiotics, such as penicillin, cephalosporin, monobactam, amongst others. Although many innovative therapies have been developed to combat this pathogen, including combination of therapies, including improvements on existing antibiotics, and then even the use of nanoparticles and antimicrobial peptides. However, this insurgence persists, thus signifying the urgency in exploring novel alternative targets. So given the vital role of NAD in controlling nearly all metabolic pathways, we see that, and besides its, going by the fact that bacteria are unable to take up NAD from their environment, they only depend solely on the synthesis of NAD. So studies have shown that by targeting the biosynthesis of NAD, there's a possibility that it could be a therapeutic, it could serve as a therapeutic purposes for treatment of multi-drug resistant bacteria. So in this study, among all the enzymes involved in the biosynthesis of NAD, we are specifically looking at the enzyme NAMNAT, which is a nicotinate mononucleotide adenine transferase. It is actually, it's an enzyme that involves the adenylation of nicotinamide mononucleotide or nicotinate mononucleotide. It involves the adenylation to actually form NAD or NADA, that is adenine dinucleotide, with the release of inorganic pyrophosphate. This enzyme actually belongs to the nucleotide transferase enzyme. And what makes this enzyme very crucial and important in NAD synthesis is because bacteria are able to synthesize NAD either de novo or salvage. They either synthesize NAD by salvage or de novo. Like I said earlier, they can't take up NAD from their host environment. So they depend on synthesis. But this enzyme, the both pathways, either the de novo or the salvage synthesis, meet at the reaction which is catalyzed by the NAMNAT enzyme. So this enzyme is actually very crucial for the synthesis of NAD. Then it's also important to know that this enzyme is characterized by highly conserved HTXGH signature motif and the presence of Roseman fold. Besides, this enzyme actually is a metal activated enzyme. It requires the magnesium metal in order to function effectively. Although studies have also shown that besides magnesium, a couple of divalent metals have also shown activity with this enzyme. I would also like to say that the enzyme is also a globular protein. It comprises of about 160 to 400 amino acids, and it has subunit mass which ranges between 20 to 50 kilodalton, and it exists majorly as homodimer, although we have a couple of NAMNAT that has been showed to be monomers. One thing that is very important to know is that this enzyme is actually very conserved in almost all species. However, each of these enzymes and their isoform, they exhibit different structural and catalytic properties that reflect their species, their specificity, and other functions acquired. So until now, NAMNAT in Klebsiella pneumonia has not, there's very limited information that has been established on NAMNAT from Klebsiella pneumonia. Hence, this study intends to describe the biophysical structure of recombinant NAMNAT from Klebsiella pneumonia using empirical and computational studies. And this I was able to achieve by first of all expressing and purifying. So the experimental aspect was carried out by starting with the expression and purification of the protein. And the expression conditions, the protein was expressed in E. coli expression, E. coli T7 expression system using PET28 vector construct. And the expression was, overexpression was induced with 0.5 millimolar IPTG. And incubation was at 30 degrees Celsius for 6 hours. Then we see that this condition were actually successful, given that from the lysates, we can see the tick band showing that there's an overexpression, which is also consistent in the supernatant and in the pellet. Then this protein was, the N terminal of this protein was actually tagged with histidine tag in order to to enable its purification using nickel ion immobilized metal affinity chromatography. And from the result we have here, we can see that the purification was actually successful. From the final wash, you see there is no sample then, but then at the elute, we could see that the protein was successfully purified using 350 millimolar imidazole. Okay, so I further went to confirm if the enzyme is active by carrying out a dual enzyme catalyzed reaction. Like I said from the introduction, NAMNAT actually is a dual enzyme catalyzed, is a dual substrate enzyme. So it can either use NMN, which is a nicotinamide mononucleotide, or nicotinate mononucleotide. So it can use either of the substrates to generate NAD. So NAMNAT, in this experiment, we actually use NMN with ATP, and then the product NAD was then further converted to NADH, which was quantified spectrophotometrically at 340 nanometer. And furthermore, I went to check the pH activity of the enzyme. It is common between NAMNAT enzyme. They have varying pH activity. However, it's at least they have an optimum pH activity between 7 and 9. So for KP-NAMNAT, the optimum activity was at pH 8, but it has a pH range between 7 and 9. The enzyme seems very active. So since the enzyme involves the use of divalent cations, further analysis was carried out to see if KP-NAMNAT is going to have preference for other divalent metal outside the magnesium ion. And from the results we have here, it's obvious that KP-NAMNAT prefers magnesium ion compared to other metals, although nickel ion was it also showed activity, good activity with nickel ion. However, it prefers magnesium ion. So I further went to characterize the secondary structure to see how the secondary structural content of the protein is. And then using 5 UV circular dichroism, which measures the degree of electricity, the degree of right-handed and left-handed electricity, and of the backbones of the protein. And from the spectrum we have here, we can see that a peak at 190 nanometers and a trough at 220 nanometers and 208 are indication that the protein has more of alpha helix. And then this was confirmed by subjecting the data to by submitting the data to dicroweb analysis, and then the data shows that, sorry about that, the data shows that the protein is actually alpha helical with having about 57% alpha helix, and this also correlates with the predicted structure from Swiss model. Okay, furthermore, the quaternary structure of the protein was analyzed to see how the protein interacts with its ligands. And then this was carried out using ANS, that is 8-anilinone naphthalene sulfonic acid, ANS. This, the ANS fluorescence dye actually works by excitation of this. This fluorescence works by when it binds to the hydrophobic pockets of a protein, its fluorescence intensity increases. And then there's actually a shift in the wavelength. And in a situation where the protein is not bound to an hydrophobic pocket of a protein, there's actually a decrease in fluorescence intensity of the protein. So from the results we have, we can see that the binding there was binding between ANS and the protein. The black spectrum shows the protein, the native KP-NAMNAT protein, and then in the presence of NMN, there was slight displacement of ANS by the decrease in fluorescence intensity. Also in the presence of ATP and NAD, we see that there was decrease actually in fluorescence intensity of ANS, suggesting that the binding of those ligands actually displaced ANS, which shows the possibility that this ANS might actually be binding to possibly the binding site of this protein. Another thing to note is that the binding of these ligands to this protein did not actually induce any conformational change. So there was actually a decrease in fluorescent intensity, suggesting that there was displacement of ANS, but there was no shift in the wavelength, in the maximum emission wavelength. Furthermore, the continuous structure of the protein was actually characterized, and from the spectrum we have here, it's obvious that the protein, the protein came out at about 20 kilodalton, and this corresponds to monomeric structure, although the 25 kilodalton is somehow smaller compared to the expected size we're actually looking out for, because we estimated it to be about 25 kilodalton. However, it came out smaller, which suggests the possibility that the protein is very globular in structure, such that it's so compact, such that it is retained so much in the matrix. So furthermore, it also shows that we can see that there's a small shoulder spectrum, a small spectrum here, which suggests the possibility that the protein also exists in oligomeric state. This peak was amounted to about 50 kilodalton. So that there's a possibility that the protein, the protein possibly dimerizes to maybe carry out its function. So after the experimental study, I further went to do some computational study in order to augment the empirical study, and to also provide more insight into the molecular and atomic structure of the protein. And then the homology, because Kepinamnat is a novel protein, and there's no structure yet for the protein, so we had to carry out an homology modeling using Swiss model. And the crystal structure of E. coli namnat showed demonstrated about 86% sequence identity with the sequence of KP namnat. And then this E. coli namnat was used to actually model the 3D structure for KP namnat that was used for this experiment, in order to validate that the structure we have is reliable and can produce a result that can be inferred. So Ramachandra plot statistic was actually carried out, and then we could see from the plot, from the statistic summary we have here, that most of the about 94% of the residues are in the most favored region, while zero residue is in the disallowed region, which shows that the model we have is actually a good one and can be used. Furthermore, we can see from the structural alignment of KP namnat and E. coli namnat that the protein, we can see that the RMSD, that's the deviation between the two structures, is very minimal, just about 0.082 deviation in their structure, which shows that this structure is reliable for the computational study we are about to carry out. So an induced fit docking was carried out in order to gain insight into the binding site of the protein and the protein interaction with the ligands, to see if the ligands actually bind at the same site, and to also understand the stability of the protein. So an induced fit docking was carried out, and this docking was accomplished via it was executed in Strontium Maestro version 12.2 algorithm, which involves the use of OPLS 2005 force field and in an implicit solvent model. And the results we have here shows that all the ligands actually bind at the same site. As you can see from the 3D structure, the ligands bind at the same site. And basically, induced fit docking also shows us some different poses. We also see that the poses that okay, the A shows the A represents KP namnat in the presence of ATP. Then this is actually KP namnat in the presence of NMN, and then KP namnat when bound to NAD. And we could see that basically there's the forces that stabilizes them is actually is majorly van der Waals forces and the hydrogen bonds that actually stabilizes the binding of this ligand to the site. So furthermore, MD simulation of the top poses of this induced fit docking was actually subjected to MD simulation, and then a post-dynamic analysis was also carried out. And then from the results of the analysis, the result of the trajectories, we're able to analyze the RMSD, that's the root mean square deviation of the C atoms of the protein over 100 nanoseconds. Then also we checked the RMSF of the protein, and then we also did the radius of gyration and also see the RMSD of the ligands. So the RMSD of the protein with respect to the protein, we could see that the black spectrum, the black profile rather, shows that's for the APO. Then the blue is in the presence of NAD, the green when the protein is bound to NMN, and the pink when the protein is bound to ATP. We see that just to remind us that the protein is a metal activated enzyme. So the experiment was actually carried out both in the presence and in the absence of magnesium. And then in the absence of magnesium, we see that there were no much deviation from the protein when the APO structure, that's the native protein, when bound to the ligands, there was not much deviation. However, in the presence of magnesium, we see that there was deviation after 40 nanoseconds in the structure of the protein in the presence of ATP. And then, although this deviation is actually within range, because deviation of small globular protein within 1 to 3 angstroms are actually acceptable. Though there was a deviation, or there's a deviation, but then the deviation is still within an acceptable range. Furthermore, the root mean square fluctuation for the side chains of the protein was also analyzed with respect to the protein. And then we could see that the A shows the C alpha atom, that's the RMSF of the C alpha atom, and while the B represents the side chain of the protein residues. So this is plotted against the residues of the protein, and then we see that in the absence of magnesium, this is what the profile looks like. However, in the presence of magnesium, we could see that this pink profile, which stands for ATP, it's obvious that there's fluctuation in the side chain of the protein in the presence of magnesium. When magnesium is bound to the protein in the presence of when ATP is bound to the protein in the presence of magnesium. Also, this is also consistent, as we can see from these residues, also that there's actually fluctuation which were not in the absence of magnesium, but then there's fluctuation within the between the 120 to 150 residues is actually fluctuation. So furthermore, the radius.
Wrap up your talk.
Sorry?
Yeah, could you please wrap up your talk?
Okay, yes, please. All right, so I think I'm missing a slide, but then the radius of gyration also showed that there's fluctuation in the presence of magnesium. So there was a massive deviation in the presence of magnesium for ATP. So checking the RMSF of the ligand, ATP, and then we could see that ATP is actually dynamic amongst all the ligands. It's fluctuating between 2 and 4, which is higher compared to other. So we could infer from here that the binding of ATP to the protein is actually the dynamics of the protein is actually dependent on the binding of ATP, and because magnesium stimulates the binding of this actually confirms to us that magnesium is actually needed for the binding of this protein, is actually required by ATP in order for this protein to be active. So I would like to acknowledge my supervisor, Dr. Achilonu, and then the Protein and Structure Function Research Unit in BITS. Thank you very much for your time.
Hey, thank you very much, Olamide, for a very interesting talk. So the floor is now open for questions. Okay, I have a question from my side. So at some point you discussed the docking mechanism, and you mentioned that it was an induced fit mechanism. So why did you opted for that mechanism and not a conformational selection mechanism? I assume you know between the difference between the induced fit and the conformational selection models.
Yes. Okay, so the reason why I actually went for the induced fit mechanism is because basically, well, let me just say I decided to go for the induced fit mechanism because though that is actually the software we actually have that is available, and then I just wanted to lay my hands on the computational aspect and then just to see what it's going to look like since it's a novel protein.
Okay, yeah, it's basically a question of when does the conformational change take place? So before docking or just after docking?
Sorry?
So, yeah, so the main difference between these two mechanisms is the stage when the conformational change takes place.
Okay.
Induced fit says it takes place just after docking, and conformational selection just before docking. So do you have information about that?
I should. Sorry, I didn't get the question.
Yeah, so when an enzyme docks, there should be a conformational change that takes place, even if it's a small change. So do you know when this conformational change takes place, before or after docking?
Okay, before or after docking, the conformational change takes place. It's going to be after docking.
Okay. Okay, yeah, then that fits the induced fit mechanism very well.
Yes.
Okay, thank you. Yeah, so is there another question from the audience?
Okay.
Yeah, I got a message that Meniko Ritsi had a few questions to ask, but apparently he had to leave before the end of your talk. So I will put you in touch with him because I don't see him being around anymore. Oh yeah, he's still there. Yeah, so the floor is open for questions. Please do ask any questions that you have.
No, it's actually going to be a number of questions because I'm working on the same enzyme. So I think the best would be, is basically the comparison of the human counterpart. So possibly the best would be to be in direct touch by emails. And anyway, a very quick one will be, I mean, how your simulation is actually taking into account the different specificity for nicotinic acid mononucleotide nucleotide for the bacterial enzyme against nicotinamide mononucleotide for the human enzyme in terms of drug discovery. But as I said, I sent a message, so we can also discuss later on because I think we're running out of time. But the major point is selectivity of action of design inhibitors. Thank you.
Okay. Thank you very much for that.
Yeah, did you want to comment on that, Olamide?
Okay, if I get him right, I think he's talking about the specificity. That's the difference between the human NAMNAT and bacterial NAMNAT. I'm not sure I got that right.
Exactly. That's exactly the point. Yeah.
Oh, okay. Yeah. Okay. Well, just because of time, I couldn't really divert much into it. But of course, there are actually differences. And for my research, I was actually working with two different proteins, two different bacterial proteins, and even though despite their bacterial proteins and they have similarities, higher similarities compared to human NAMNAT, it's obvious that even those bacteria, the NAMNAT, they have different structures and features. So it's obvious that it's obvious that I'm trying to tell to construct the sentence. Pardon me about that, please. sorry, please, can I just keep that, please? Hello?
Hello?
Yes, please, can you hear me?
Yeah, yeah.
All right, yeah, I said, please, can you pardon me? Let me just skip that.
Yeah, no problem. I mean, we can also be in contact by emails because they sent a message with my emails, so we can also discuss later on.
Okay, thank you.
Yeah, pleasure.
Okay, yes, let's do that. I will then put the two of you in touch. Okay, is there any last pressing question? Okay, if not, I would just like to comment that, yeah, for a master's project, this was really remarkable, and I would like to commend you for this.
Thank you very much.
This is really a great project, and yeah, also a very nice talk. So thank you very much for this presentation, and let's also give an applause to all the speakers of this session. So a virtual applause. Yeah, so a lot of different systems have been shown, different biomolecules, proteins, and so on. So that has been very interesting to see all this diversity. And yeah, so this concludes today's session on the molecular biophysics. And please do have a look at the program for the rest of the week. So tomorrow morning, we are going to continue with X-ray scattering, spectroscopy, and radiation in biophysics, starting at 10 o'clock Central African time. And I hope to see all of you again during the next of this conference. So thank you very much for everyone's participation, and yes, we see you then later. Goodbye.
Thank you, everyone. We'll see you tomorrow.
Thank you. Thank you very much.